yang Good morning, sir.
Good morning. How are you?
Fine, sir.
Hello. Good morning, madam. Srikala, madam. Good morning, madam.
Sir, good morning, sir. Shall I start now, sir?
Yes, madam. You can start, madam.
Okay, sir. A very good morning to all. Hearty welcome to the second day of International Econference on recent trends in pharmaceutical research with a theme, Research Innovations Leading to Intellectual Property Generation in Drug Discovery and Development. Organized by Nirmala Innovations and Incubation Center, Nirmala College of Pharmacy, Mangalgiri, Guntur, Andhra Pradesh. In association with University of Utah, College of Pharmacy, USA, and Thermo Fisher Scientific, Canada, Faculty of Pharmacy, Kalinga University, Raipur, Journal of Cardiovascular Disease Research, and Research Journal of Pharmaceutical Technology. We completed first day of International E-Conference successfully with the following achievements. Around 79 colleges participated in the International E-Conference yesterday, with 645 registrations and 145 articles accepted for publication. Around 150 oral and e-poster presentations completed. With the triumph of first day, we will start second day of International E-Conference with the same zeal and commitment towards the success. Thank you all. Now, I would like to invite Mrs. Supriya ma'am to continue the session.
Thank you, Mrs. Srikala ma'am. A very good morning to all the delegates. Once again, hearty welcome to the Day 2 International E-Conference on Recent Trends in Pharmaceutical Research. Research innovation leading to the intellectual property generation in drug discovery and the development, organizing by Nirmala Innovation and the Incubation Center in association with the University of Utah, USA, Thermo Fisher Scientific, Canada, Faculty of Pharmacy, Kalinga University, Journal of Cardiovascular Disease Research, and Research Journal of Pharmaceutical Technology. Let us summarize the yesterday's scientific session before move on to the today's session. In session 1, Dr. A. Buchiraju sir gave an outstanding presentation on evaluation of clinical studies. Sir has given detailed information on descriptive studies, observational studies, and the experimental studies. Sir also stated the importance of controlled experimental design and types of controlled experimental designs. In session 2, Mrs. Nisha Madhwa, Amira Patent Technology, New Delhi, gave an excellent presentation on intellectual property rights and on the patents. Ma'am gave detailed information on importance of patent, filing of the patent, and main elements of the patent application. Ma'am also gave the information on foreign patent filing and the importance of startups. Today, the program starts with the scientific session 3 up to 11:30, followed by e-poster and the oral presentations up to 12:45 p.m. in the morning schedule. And we have valedictory program in the afternoon schedule, which starts at a sharp 2 p.m. And coming to the today's session, scientific session 3, resource person, Dr. Benjamin Johnson, Thermo Fisher Scientific Limited, senior formulation technologist, Canada. Scientific session 3, chairperson, Dr. N. Bujnaidu sir, HOD, Department of Pharmaceutics, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada, Andhra Pradesh. Before inviting the chairperson, I request the delegates to post the queries in the chat box. At the end of this session, all the queries will be clarified by our resource person. Now, this is the time to invite today's chairperson, Dr. N. Bujnaidu sir. I'm very much delighted to introduce session 3 chairperson, Dr. N. Bujnaidu sir. Sir is currently working as professor and HOD in the Department of College of Siddhartha College of Pharmaceutical Sciences, Vijayawada. Sir worked as Research Associate, College of Pharmacy, University of Kentucky, USA. Sir has been completed his PDF from College of Pharmacy, University of Kentucky, USA. Sir has received 5 research grants from various governing bodies like DST, AICT, Ministry of Science and Higher Education of Government of Poland, and under the Indian-South Africa Agreement on Science and Technology Cooperation Program. Sir has around 5 Indian and the US patents. Sir also acted as principal scientist in UPenn Therapeutics, USA. Sir has acted as principal scientist And Sir has received Best PhD Thesis Award and Gold Medal by Andhra University. Sir has developed Valaclor, a topical mecloretamine gel, and was approved by FDA, which is available now. It is available commercially in the USA. Now, I would like to invite Dr. N. Bushnaidu, sir, to chair the session 3. Sir, please start the session. Sir. I invite Bushnaidu, sir, to start the session 3.
Yeah, thank you, Supriya. Am I audible?
Yes, sir, you are audible, sir.
Okay. Thank you, Supriya, for the introduction. And good morning, organizers, participants, and today's speaker, Benjamin Jason from Canada. So I appreciate the Nirmala College of Pharmacy in continuously organizing the several national and international seminars, e-conferences, webinars, etc. And when coming to the today's conference, International E-Conference on Recent Trends in Pharmaceutical Research, with a theme or concept on the research innovations leading to IPR generation, especially in drug discovery and development, which is the need of the hour around the globe. And I appreciate the Nirmala College management and principal taking efforts in organizing these up-to-date technology seminars or conferences online so that the students and the community will get benefited. And when coming to the today's session by Mr. Benjamin Jason, who graduated from Gulbarga University with B.Pharm degree in the 2000 and Master of Science in Bioinformatics from Satyabama Institute of Technology in 2006. And also he had PG Diploma in Pharmaceutical Research and Development Technology in 2017. And then he worked in several multinational companies like Park Davis, and also he worked as a faculty member in Bioinformatics and Bharat University, Chennai, and a student coordinator also where he worked as in Satyabama University as a lecturer and assistant professor. And later on, he moved to Canada, I think in the year 2016, and he worked as an implant trainee in apotex and production technician in apotex, and also as a part-time faculty member in Durham College. And presently, he is working as a senior formulation technologist in Thermo Fisher Scientific with Toronto. And today, he is going to talk on the role of bioinformatics in pharmaceutics. So the good thing that Mr. Benjamin did is he has diverted from pharmacy profession, which is a basic degree he had, onto the bioinformatics sector. And presently, this is the need of the hour in the pharmacy education. So pharmacy education means not only you just acquire the pharmaceutical skills, you need to acquire the other technical skills in the other field like biomedical sciences, bioinformatics, molecular sciences, molecular biology, so that you get plenty of chances in different fields of research and companies where you can have an interdisciplinary skills so that you can shine and grow further in our career growth. And my sincere advice to the budding pharmacists is don't end up with the pharmacy, acquire the skills in other fields of science like Benjamin did in the bioinformatics. And he is going to talk today especially on the concept of role of bioinformatics and pharmaceutics. And once again, I appreciate the Nirmala College management and Principal Dr. Rahman and the staff and the students of the college and the participants of the program. And I hereby welcome Benjamin Jason, please. Thank you.
Good morning. Can you hear me?
Yeah, please go ahead.
Thank you, sir, for the introduction. And thank you for the management of the Nirmala College of Pharmaceutical Sciences for giving me this opportunity to share my knowledge and experience. So I would like to switch to my presentation here. John. Okay. Okay. Can you see the slide, sir?
No.
No? Oh, sorry. Okay, screen.
I think you have to share your screen.
Can you see it now?
No.
No, sir.
Oh, sorry. Two. I think it's
Yeah.
yes. Okay.
Yes.
Thank you. Yes, thank you. So yes, I'm now going to talk on the role of mathematics in pharmaceutical sciences, especially with the concept of research. Uh, as I was introduced by Sir with pharmacy background and then switched to bioinformatics, I never thought that this will be a good combination before, but once I was into that and gone uh, through this, definitely there is a great application, and we will see that in a bit through this presentation. So I came here in 2015, and yeah, I have taken a research and development program, technology PhD diploma, almost after 15 years. So I'm back again into pharmaceutical field, but bioinformatics is really a great combination always to help in the research. So in my presentation, I'm going to talk topics and just an introduction about bioinformatics and what are the branches in bioinformatics, and little more on the topic drug designing and discovery, where bioinformatics plays a good role in downsizing the time or the cost, manpower, and material management and all that stuff. And then a brief time on databases, softwares, and then some resources where if students are interested, maybe that will be helpful. And then I'll be winding up with the applications of bioinformatics. So the introduction, by definition, uh, bio is life and informatics is something to do with IT. But as the research or a lot of data started accumulating years back, there's a lot of data where people can't manage or contain it, or retrieving the data or working with the data which is already collected was really tiresome and time consuming. And when the computers have just come into the markets, they started using the computers to store the data. and use the data for the analysis. So it is from that time they started using the computers. So by definition, bioinformatics, it's a science of collecting, classifying, storing, and analyzing large and complex biological and biochemical data using computers. So say, for example, the DNA sequence, it is huge. And working with the protein sequences or the protein structure, uh, the data is so huge, and the protein structural data can also be generated in computers and it can be stored. So all this data can be collected and stored, and it's not just storing alone, but you can start working with the data. That is where now bioinformatics can be applied in drug discovery. So that is what the history says: large data, and then the problem of storage, and it also helps now in data analysis. So there are two major areas in bioinformatics actually. If somebody like I assume I'm talking with all the B.Pharm or M.Pharm students here. So as I'm also from pharmacy background with bioinformatics, we have two areas. The first one is bioinformaticists, who develops an algorithm for the problem using any programming language. It can be Python, Java, or R script, or Perl, and so on. And even the databases like Oracle or whatever. So bioinformatics is the one who actually develops a tool or a software which can be used in actually solving a problem. So there comes the bioinformaticians. So sometimes I myself, I am not a coding guy. I don't develop programs, but I use the software. So I fall in bioinformatician. See, sometimes if you're very good in programming, you can be both. So, but coming to Canada, I see that a lot of hospitals who work in any kind of data, most of the, like I see cancer patients, when their DNA or their target protein is sequenced or the structure, they they work with a bioinformaticist or a bioinformatician specifically for that particular patient. So they you have a lot of opportunities for the bioinformaticians or bioinformatician uh, across the world now. And what are the main branches in bioinformatics? Bioinformatics has basically you start with two things. One is working with the genome data, where the DNA, RNA sequences are being stored, and then working with them, and then the protein data, the sequence and the structure or the interactions of proteins among themselves. So they are widely called as genomics and proteomics. And from here, you have a lot of other sub branches, transcriptome analysis, where mRNA expression levels can be determined, and cheminformatics, working with the chemical stuff or quantitative structure activity relationship, or working with a drug molecule, screening it virtually. And then the last one is medical and health informatics. Basically, actually, these are just few words which I mentioned here, but most of the combinations with IT from the bio field or pharmaceutical field, you have everywhere computers involved. So there's a lot of scope once someone is trained into bioinformatics. Now, but today, drug discovery and drug design and discovery is the most relevant topic for the pharmaceutical background students and those who are planning or pursuing in a career like that. As we know, traditionally speaking, the drug discovery takes a long journey. Uh, on an average, it should take uh, eight 8 years or 10 years or even more than that in a conventional way. starting with a drug molecule, and I mean the most potential drug molecule, screening them, and at last you get starting from 10,000 to 20,000 compounds, and you narrow down to just one, and then this will be the most probable lead molecule, and then it goes on for the preclinical studies, clinical studies, or the formulation studies, and establishing um, the formula, and then goes into the market. So it's a tedious process and it takes a long journey, but yeah, that is how it's going on until now. But uh, there are some drugs which are already into the market, which are completely designed and developed through bioinformatics and synthesized later in the labs, and then formulated and clinical studies, preclinical, clinical, and brought into the market. But there's also another way where bioinformatics comes half the way, or at the initial stage, to speed up all this, the 10,000 or 20,000. It can be when, I mean, unlimited. You have a lot of databases where all these chemicals or the most potential drug molecules which are stored, you take your own molecule and just search in that database, you get a lot of hits out of it, and you can start working from there. And then it passes on. So it can be solely on bioinformatics, or it can just be the both conventional and bioinformatics bridged together, which speeds up the work. So, and it's not only that, a lot of money is invested, billions of dollars. So it takes 10 to 12 years or too much of money. Now, what exactly is drug designing? Computer aided drug designing, it's also called CAD. There are basically two types. One is receptor based drug design, and the other one is ligand based drug design. Receptor based drug design, basically it's receptor is nothing but the protein that's the target of disease, where we try to mutate or degenerate with the help of a drug molecule. So so the first one is starting from the protein, that is the protein of interest of the disease, that is the receptor, and then go and design the drug molecule. Okay. The second one is you have the drug molecule, or you have potential drug molecule, but from there you design it where you can have a target molecule or a target protein or a receptor. So it is on the both sides, from the target to the drug, and or from the drug to the target. So this is again the same slide. It's the same picture, but yeah. Um, the first part you see the structure based, and the second one is the ligand based. So when you say the structure based, it is the target structure protein. Um, sorry. Um, and here now bioinformatics comes in play actually at the structural level, both in ligand or in the protein. Okay, so conventional way, yeah, we can work with sequences, but when it comes to structure, computer is the best source where you can model the structure. You have a three-dimensional structure. You can program it with all the coordinates. Okay, so all the three coordinates, and then work with the structure. So that makes things so faster and easier uh, computationally. So target structure on the structure based is nothing but working with the protein structure. When you see the protein structures, you don't have a lot of structures in the databases. You have tons of sequences, but not the structures of every sequence. So there is a lot of work required, even um, getting the structures of the proteins. So working with this target structure, Again, then once your structure is ready, your ligand of interest can be worked with the target structure. It's you use this protein as a lock and ligand as a key. So it is like lock and key hypothesis. So that's called, the process is called as docking. Okay, so the ligand docking with the task structure, or it can be de novo design. You don't have any ligand, but when you work with the protein structure, you can go with the most potential active sites or the cavities in the protein and see what are the atoms or the molecules involved there, and you can design your own ligand molecule. Ligand is nothing but the most potential chemical compound. It could be a drug molecule eventually once it is successful. So these are the two things after target structure. And then once this is done, then we work with something called MD, it's molecular dynamics, working with all the forces. Once the ligand or the most potential drug molecule is fused with the protein structure, we see the energies or the forces, how stable it is, how long it can, I mean, how much time it takes to denature the protein. Okay, and then you also called something called as pharmacophore modeling. Pharmacophore is nothing but the site, the functional site of your ligand. That's the site. I mean, the functional part of your drug molecule, which is actually uh, binding with the protein cavity and then denaturing the protein which is causing the disease. Okay, so the other side is the ligand based computer aided drug designing, where you have the ligand structure information, and from there you screen virtually, or you do the quantitative or qualitative structure activity relationship with the other compounds, and then you model. You also have the modeling of pharmacophore here again. So in both the ways, you're going to optimize the lead molecule, and then you get the most potential drug molecule. So that is how you go until the receptor, or from the receptor to the drug, or from the drug to the receptor. So receptor is the target, as I said. Usually it is a protein. In most cases, it is protein. And now, as I said, the protein structures, not necessarily all the proteins has the structures. Or say, for example, now any virus or any new diseases, the proteins sequence and its structure has to be elucidated, or it has to be determined. So now here it comes the central dogma, the structure determination of receptor, the central dogma in the biology or the biological sciences. It is starting from the DNA, it transcribes to RNA, and from there, it I mean, from DNA it translates to RNA, and from uh, RNA it translates again to a protein molecule. So now uh, here there is a lot of work which is usually available in the research area, where there's a lot of need for uh, structures to be elucidated, and a lot of jobs obviously um, available in this area. And from there it goes with, as I said, drug molecule binds to the protein causing the biological effects. And yeah, so lock and key system is the same thing, the protein and the ligand molecule. And virtual high throughput screening, here the protein targets are screened against the databases of small molecule compounds to see which molecule binds strongly to the target. So you get the most potential compound for your protein target, which can strongly, you have a lot of screening techniques. And then it's the same thing again, determining the structure of the host, building a model of the binding site, and searching in the database for the new ligands, and dock this ligand to the binding site of the host, predicting the binding constants or activity, the forces or the the energies, and then it comes again with the conventional method. So it has to be synthesized again. So working with all this, you develop a molecule, and from there, at last, it has to come back to the lab, and we have to synthesize the molecule. Okay. So this is again the same thing what I just explained about ligand-based drug designing. Ligand is nothing but a molecule that binds to another molecule, and it's usually the large molecule because it's going to be a protein molecule, and it's massive. It's a macromolecule. So drugs are considered to bind to receptors, and any chemicals that bind to receptors are usually termed ligands. So ligands interact with proteins in target cells, which are cells that are affected by chemical signal. These proteins are also called receptors. So we are searching for a lead compound or an active ligand, and then if there is any modification of known drugs, sometimes we need not go for a new drug. You can also work with the existing drugs, where you play with the pharmacoorigin and the functional part of it. You can screen and get multiple other functional groups where it can be a potential drug for the new target. And the third way, as pharmacognosy, naturally also you can modify or whatever is there naturally. From there also you can work in bioinformatics, or you can modify whatever is there in nature to design the compound with the desired properties and to assess its therapeutic effects and theoretically. So this is just a picture how the ligand works with the receptor. So you have the databases and modeling involved, designing and all stuff. At last, you get the ligand. The receptor, that is nothing but your protein, and then you have the ligand molecule, and these ligands are nothing but your drug molecule, which are very small, and they dock with the receptor, and that's called docking. And you're going to see what molecules are involved. What atoms are involved, and going to check how stable it is. And the advantages of drug design is it's cost effective, and it is time saving, and it's very fast, and it's sometimes you have a lot of automation process too. Say for example, virtual screening, literally speaking, manually if you sit and work, it takes a lot of time, but if you're doing a virtual screening, it is an automation process. And then can obtain an idea about drug receptor interaction patterns. So and it minimizes synthetic and biological testing efforts, minimize chances of failures in the final phase. Even if it before it goes to the final phase, right at the in silico. This is called in silico working with computers. You can narrow down to a point, okay, whether this can be a most potential drug or not. And I forgot to mention here, you can also check the toxicological studies too, whether this drug molecule has the potential, the potency or ability or capability of binding with other proteins, which are not supposed to happen. And yeah, even that can be an advantage in computer drug designing. So now coming to the databases, these are the main tools. Without databases, we don't have bioinformatics. We can't do anything. So databases are our backbones. So databases are important tools in assisting scientists to analyze and explain a host of biological phenomena from sequence and structure of biomolecules and their interaction. So as I said, we have DNA databases where the sequences of DNA has been stored, and you also have databases of proteins, sequence, and their structures. So you have sequence databases. You also have structural databases, and you also have something called secondary databases where the structures which are not completely three-dimensional structures. As protein, you have primary structures, and then at last it conforms to a three-dimensional structure. So you have all these databases available. So biological databases are libraries of life sciences information collecting from scientific experiments, published literature, high throughput experiment technology, and computational analysis. It's nothing but like whoever is doing any research working with the DNA or a protein sequence and its structure, when they publish or they submit to these databases, it enters into that database, and anyone can access that, and they are all free, and you can download and work with them. And you also have chemical databases specifically designed to store chemical information or the structures or all the published uh, chemical compounds. And now coming to biological databases, you have primary databases where they are organized user-friendly gateways to the huge amount of biological data produced by researchers around the world. And the secondary databases comprise the data derived from analyzing entries in primary. So secondary is the work generated from the data which is from the primary databases. So primary is mostly sequences, and secondary you have uh, sequences and also the structural data or the interaction or any kind of output from the data, the work done from the data in primary databases. So the secondary databases comprise data derived from analyzing entries in the primary databases. In most cases, they also provide tools to investigate further the genes and proteins. So you can work with these genes and proteins, gene of interest or gene. I just want to tell one very important difference. Some genome and gene, they are not different, but they are not same. Genome is nothing but the complete DNA sequence of an organism, but gene is one functional portion or region in a genome. So gene is a part of it. It is not the complete genome. So genome is the complete DNA, but gene is the area where it translates to an mRNA or a RNA and transcribes to a mRNA, I mean tRNA and mRNA, and then translates to a protein molecule. So gene is the one which actually codes a protein. So we investigate gene of interest like cancer treatments or um, uh, some diseases um, where a drug molecule is so specific with the gene, so a drug molecule can be designed with respect to gene. And this is where actually a concept of personalized drug therapy, it's also a future therapy system where uh, chances of making a drug molecule just for you and me, it is like tailor-made medicine specific to an individual. So working with genes, and then working with proteins. They work by analyzing pre-existing data, for example, all protein sequences ever submitted, or conceptual translation of all the nucleotide sequence, and collect alongside, when possible, information about the function of that sequence. So when we say function, it is nothing but the structure of a protein. So until it, the DNA codes to a protein structure, it cannot function. It cannot show its action. So the action starts when the protein. So when you say the disease showing its symptoms, actually the protein which is released from that particular gene in that particular DNA, it starts giving problem. So and that is the gene of our interest, or the protein of our interest. So the therapy can be designed with the gene and the protein. And there are 3 nucleotide repositories or primary databases for the submission of nucleotide and genome sequences. And first is NCBI, and it's called GenBank, hosted by National Center of Biotechnology Information. So if you just Google up NCBI, it opens up the database NCBI, and you can go through a lot of sequences which have been submitted by researchers. And you also have EMBL. It's a European Molecular Biology Laboratories, and there also you have a repository, you have a lot of data. And the third one, it is by Japan. It is called DNA Data Bank of Japan, DDBJ. So these are the 3 main nucleotide repositories. So genomic databases, there are 3 nucleotide reports, as I said, and protein databases, you have something called as UniProt and SwissProt, and these are the most comprehensive source of information on proteins in protein sequence databases. And you also have, as I said, protein has sequences and structures. So you have structural databases, protein data bank, PDB, and you also have structural classification of protein, scope. So you have genomic databases, you have protein sequences databases, and you have protein structural databases. This is just one of the snapshots for the PDB and UniProt. And next follows the chemical databases. I think this is the area of interest where a pharmaceutical, and even if you don't want to learn bioinformatics, still you can use these databases, and they're free. PubChem is a database of chemical molecules and their activities against biological assays. The system is maintained by National, it's again NCBI. So you can just go around and see like what things can help you in your research. And you also have drug bank database, is a comprehensive, freely accessible online database containing information of drugs and drug targets. And you have ChemSpider, and you also have a chemical CHEMBL. It's a European chemical database, manually curated chemical database of bioactive molecules with drug-like properties. And you also have KEG ligand, or KEG database. It's a huge, and it also has tools built in where you can screen, you can design and work so on. It is a composite database named for compound glycan reaction. You have different sub databases where whose entries are identified. And now comes the software. We have softwares both commercially made, commercially are being sold to the pharmaceutical companies, and they use them working with designing dynamics or docking, whatever. And you also have some small free software. Maybe you can download ChemSketch by ACD Labs. It's a molecular modeling program used to create and modify images of chemical structures. you can just design your structures, or you have a structure and you can load it and modify it. Okay. Or now coming to the commercial softwares. One of the huge softwares is, I mean, Schrodinger, the one which I listed number 2. It has a discovery informatics suite. It has everything in it. It has sequences, it has structures, it has all the other analytical or the tools where you can work with the structures, docking, and working with molecular modeling, designing. Everything is included in this. And Autodock is especially for docking, working with the receptor and the ligand, and so on. And lastly, the applications of bioinformatics. Application of bioinformatics, you have microbial genome applications, molecular medicine, personalized medicine. I just spoke a little bit when I was talking on genes. Personalized medicine is like taking an individual's whole gene DNA sequence, or yes, and then finding the genes and or the gene of interest and translating it to the protein structure, and then making a medicine for that particular gene and protein. Then preventive medicine, gene therapy, drug development, antibiotic resistance, waste cleanup biotechnology, climate change studies, and alternative energy resources, crop improvement, forensic analysis, bioweapon creation, insect resistance, improve nutritional quality, development of drought resistant varieties in crop, and um, veterinary science. And thank you so much for being patient. And yeah, that's all from my side. And
Thank you, sir. Thank you,
you're welcome.
Jocelyn, sir, for your valuable presentation. Now, I would like to invite Dr. N. Bushnaidu, sir, to conclude the session.
Thank you, Benjamin. That is a nice presentation, especially with regarding to the bioinformatics field that is linked to the drug discovery, design, and developmental process, how it is going to be useful, and how it's going to be save the millions of dollars in terms of the drug discovery program. And I hope that now the budding pharmacists, the graduates, may recognize the importance of acquiring the skills other than the pharmacy, so that they can grow up in their career by taking you as a role model. And I hope that the student follow your route to become successful in their future endeavors. And thank you very much for the valuable talk and your time. Thank you very much.
You're welcome, sir. Thank you.
Now, this is the time to honor our chairperson, Dr. N. Bushnaidu, sir. On behalf of management of Nirmala College of Pharmacy, Utah Health Sciences, USA, Damo Fisher Scientific, and Faculty of Pharmacy, Kalinga University, sir, we request you to accept our token of gratitude in the form of e-certificate, sir. Sir, please accept our request, sir.
Thank you very much. Thank you.
Thank you, sir. And I also request today's resource person, Dr. Benjamin Johnson, sir, to accept our token of gratitude in the form of e-certificate. I request
Thank you, madam.
Benjamin, sir. Thank you, sir. Thank you, sir,
You're
for
welcome.
spending your valuable time and sharing your knowledge to our participants.
Thank you, madam. Thank you.
Thank you, sir. Now, I request the delegates to join in the given link for their e-poster and oral presentations. And there is a separate Google Meet link will send by the respective departments to start their e-poster and the oral presentations. And the competition will be continued up to 12:45 p.m. followed by the lunchtime. And in the afternoon session, we have the valedictory program. I request all the delegates to fill the feedback form and please send it back to issue the participation certificate. I'd like to inform that all the participants will receive the participation certificate and prize money for the winners of e-poster and the oral competitions of various departments along with the merit certificates. Now this is the time for the e-poster and the oral presentations. I request all the participants to join in the given link to start their e-poster and the oral presentations. And I also request, once again, I request all the participants to attend the valedictory program in the afternoon session.